This is Digital Pathology Today. Now, here's your host, Dr. Joseph Anderson.
Imagine a world without staining. In the excitement of digital pathology, we often forget that we're left with a legacy system of staining tissue pinks and purples, which is time-consuming and tissue-destructive. There's got to be a better way. Welcome to Digital Pathology Today. I'm Joe Anderson. Our guest is Dr. Gideon Ho, who co-founded HistoIndex to develop and commercialize the world's first fully automated quantitative stain-free imaging system. Under his direction as CEO, the laser-based stain-free imaging system at HistoIndex was successfully implemented in academic, clinical, and commercial research centers in major universities, hospitals, and research organizations throughout the world. Dr. Ho has multiple patents and journal publications in medical imaging, lasers, and optics to his credit. We're going to be talking about what the promise is of stain-free digital pathology and some of the technologies they've developed. What is multiphoton technology, and what is second-generation harmonics? And in terms of clinical applications, the area HistoIndex is focusing on first is NASH, nonalcoholic steatohepatitis. We're going to talk about that. What is it? What's the unmet need? How does it affect society? What are the diagnostic challenges and areas that we can improve upon compared to standard histology? Gideon Ho, welcome to the podcast.
Thank you.
Now tell us a little bit about HistoIndex. How long have you guys been around? What do you do? What's your mission? What purpose does the company serve?
Right. Well, HistoIndex, we have been around for about 11 years right now. We got incorporated in 2010. We started off from a research lab in Singapore. We spun off from the research lab into a company. And what we do mainly is to use multiphoton stain-free technology to look at tissues, biopsies of both human as well as animal tissues, mainly to look into endogenous biomarkers within the tissue, and then apply software algorithm onto these images and to extract big data out of it, mainly to look into tissues that are very fibrotic using multiphoton technology to look into it. So the whole idea of what we're trying to do is that, and when we first started off 11 years ago, this was a very new thing. The whole digital pathology space was a new thing. We wanted to set up a new standard, to basically have a new standard of how tissue diagnostics they are being done, and how would that impact in terms of diagnosis of especially fibrotic diseases, and how that will be, coming forth and using our technology, using our AI, artificial intelligence, be able to come up with, or basically help drug development company to get better drugs that works. So that's really a whole basket of ideals and mission, but mainly is really to see how we could better mankind in terms of, able to come up with better treatment in terms of for healthcare purposes. Yeah.
Stain-free multiphoton technology. Did I say that right?
Yeah, it's quite a mouthful. I kind of understand that.
So it seems like that would have a very broad application, because one of the challenges with digital pathology is maybe to reimagine the workflow or the way we handle tissue, because what we're actually doing is we're, using an old technology, H&E staining, for the most part, and then trying to retrofit that technology or, stain the tissue with H&E and then load it onto a scanner and then view things digitally, but handle the tissue the exact same way. So it seems like this application would have a very, very broad use and could totally change the way we do things. But you started in a very small area with NASH, non-alcoholic steatohepatitis. So maybe tell us, how did you get started focusing on that area? What is it about that disease that caught your attention? What is the societal impact? What challenges did you see in that relatively small area?
So when we first started off in a research lab, we were looking at mainly liver tissues, liver fibrosis, and we were looking at viral-based fibrosis, viral-based insults that basically causes fibrosis in the liver. And then slowly we evolved to look into, of course, the usual hepatitis B, hepatitis C, and then we got into fatty liver. So fatty liver, and before I talk about NASH, I need to talk about fatty liver. Fatty liver is really the accumulation of fat in the liver. In the liver itself causes a lipotoxicity that causes by the accumulation of fat. Now, that fatty liver will be okay, harmless, now until inflammation sets in and scarring sets in, and that's where you will get non-alcoholic steatohepatitis. And then with further scarring where it replaces liver cells, that's where you get cirrhotic liver. So non-alcoholic steatohepatitis is mainly the accumulation of fat plus the inflammation and the scarring. That's where you get NASH. So NASH today, about 25% of adults worldwide have fatty liver. It's closely associated with type 2 diabetes, insulin resistance, and the obesity, the epidemic itself. So 1.4 billion people worldwide, and we're talking about adults right here, out of which 30% of patients with fatty liver progress to NASH, and out of which 20% would then progress to a more severe form of liver fibrosis, which is liver cirrhosis, and that's what it is. It's a global pandemic with an unmet need for effective treatments. It's a chronic pandemic, if I can say that.
Okay,
That's yep. Go ahead.
that's amazing. 25 to 30%. I mean, that's a very large portion of the population. I think it's kind of, I don't know if you would agree, it's maybe like a silent pandemic because, in my experience, there was never much sympathy for it, or, for maybe a lot of reasons. Like it seemed like it was number one, it was kind of like a non-specific finding. Right?
Yes.
And, oh, that's just associated with obesity, or that's associated with poor health, or, something like that. But it didn't seem like it was a very specific disease and that there was much concern about finding a cure or being able to better diagnose or classify these patients. So it seemed like an unmet need, but a very large one.
Oh, yes, it is. It is indeed like a silent killer, mainly misunderstood chronic condition where it's linked to obesity. Of course it is, but once the liver get into a stage whereby it's fibrotic, even having a weight loss have very limited effect on that. So it is definitely a very big problem right here. Therefore, there are currently 90 ongoing drug trials in phase 1 to phase 3 NASH clinical trials in the United States and EU, and wanting to bring forth promising drugs to the patients with NASH. And none of the drug has been approved yet. It's currently all basically on trials right now. So it is definitely a very big global market. In fact, I can tell you it is expected to reach about $84 billion in 2029. It's growing at a CAGR of about 40% from 2018 to 2029. That's really the projection.
So from a diagnostic perspective, what were the unmet needs, or what were the areas that we could do better in? Perhaps developing a system of classification, coming up with various criteria to better classify the disease, or find histologic features that would correlate with outcomes and maybe even response to drugs. I mean, what were the areas of focus from the perspective of histopathology that you could really develop tools to help, advance the diagnosis and treatment of this disease?
Yes. So the current gold standard to diagnose NASH is still biopsy based. So it's looking at the biopsies of the NASH patients, and the way to diagnose it definitely will still be based on pathologists looking under the, using a set of categorical scale stages and scores to look into the stratification of fibrosis, as well as the activity of the fatty liver, which they are inflammation, steatosis, and ballooning, which is really a ballooning of the hepatocytes. So these are the usual way of diagnosing for NASH. But as you will know, in terms of that, it is very difficult for that to be categorical scored in a very consistent manner. And that's where digital pathology comes in, especially in the area of drug development, where you are looking into measuring the magnitude of efficacy in a drug. You will need to have a more objective and more consistent methodology and technology to measure, especially if on a continuous scale and not just on a categorical stage 0 to stage 4 kind of a scale. So the whole challenge right now is actually in the field of drug development, where how we could have a better, more sensitive way of measuring the effect of the NASH drug. And that's really what FDA is currently looking at it right now too. And I would say the whole field, the whole NASH field is trying to grapple with this particular challenge on this particular diagnostic challenge on measuring efficacy of drugs undergoing trial right now. Yeah.
Yeah, I think it really highlights what the promise is of digital pathology. I think in my mind, two things would stand out. One is, from a metabolic perspective, I think hepatocytes are incredibly complicated. There's a lot going on in terms of the metabolic machinery in the liver, and probably much or all of it is not visible to the human eye. So is there an opportunity, for molecular type analysis or different stains that can highlight various metabolic pathways? And then B, in terms of fibrosis, I mean, we can all, we have trichrome stains or other standards that human beings can use to highlight fibrosis. But in terms of measuring things and quantifying things, I think image analysis or artificial intelligence can do a much better job, particularly with these repetitive tasks that, is just beyond the ability of a human being to consistently measure and quantitate.
That's right.
So are there tools that highlight the various molecular pathways of the hepatocytes?
I think there are quite a number of non-invasive tools, but in terms of looking at really able to drill down deep into looking into what is going on within at the tissue level, of course, you will need to look into tissue diagnostics. Now, stains, there are a lot of stains that look at. There are current stains that look into just looking at inflammation and even just look at ballooning. The most, I would say, fibrosis is definitely the killer right here. Now, everything else that causes the insult is at the end of it, you will see the resultant effect in the fibrosis, and that's the killer right there. And as you have just mentioned, mesenteric stains, picosaurus stains, these are usual stains to look into fibrosis. But with every stains, there are variations. I've seen variation in stains of mesenteric as well as picosaurus stains coming from the same lab, used by the same set of technicians in the same group of technicians, and yet there are variations in the stains. And one of the challenge and limitations of that, what we see is that with variations of stains, it will affect the AI output as well. So that's the reason why we use StainFree, in short.
Yeah, so I think StainFree, has huge promise, even beyond NASH. So are you looking beyond NASH really to apply this to all cases in histopathology or many more applications?
Yes. I mean, the short answer is yes. We have been using the same technology to look into fibrosis of the liver, the kidney, the lung, bone marrow. I mean, a whole wide is easily about 20 applications, different parts of the body that we look at using the same technology. A different AI, of course, because every organ has its own specific AI algorithm that's being machine learned and being trained to look into and quantify that particular organ, the tissue from that particular organ, and even looking at fibrosis in tumors as well. So that's another big area that we're looking at in the area of cancer: triple negative breast cancer, high-grade serous ovarian cancer, liver cancer. These are the usual ones that we use our technology to look into the cancer part.
So a lot of applications.
A lot, yeah. It would take me a few hours just to talk about the various ones. Yes.
Yeah, and you mentioned AI algorithms, of course, are a big part of it, and so I'd love to get into that. So, but first, tell us maybe a little bit about the technology. So, what is multiphoton technology?
Oh, okay. I could spend 1 hour just to talk about the physics part. I think I'm going to go real high level on this. The foundation of this technology is actually known as two-photon microscopy. So what it does is that instead of having one photon commonly used in confocal microscopy, we use two photons that have higher energy levels, and they are being emitted at the same time. So you have two photons being emitted at the same time and interacting with the tissue. It results in a generation of second harmonic signals, or what we call SSG. So these second harmonic signals will be reflected from any non-central symmetrical structures like collagen fibers, and they are then detected by the photomultiplier tube, the PMT, at a wavelength of about 390 nanometer. So at the same time, transmitted photons from the two-photon emission of TPE allows us to capture information on cells. So to summarize this, you have really two simultaneous signals coming out when the two photons interact with the tissue. One is SSG, and the other one is two photon, that looks into the autofluorescence of the cells, and the other one, SSG, looks specifically at collagen fibers. I hope that really explains it in a nutshell.
Yeah, so I think for our pathologist listeners, I think we're familiar with fluorescence and the harmonics. Certainly that's kind of what we're visualizing in fluorescence studies, but you're not using stains though, is that right?
No, we're not using stains at all. All these are stain-free, so you are looking at endogenous biomarkers from the tissue without staining them and using SSG and two-photon.
Yeah. Okay, so you're exciting the photons that are naturally occurring in the tissue.
That's right.
So how it results in the SSG, or the second harmonic generation. So how specifically is the SSG applied as an endpoint or a marker, say in trials looking at NASH? Maybe could you explain how that would work?
So what SSG does is to allow the collagen fibers to become visible, even though, so in unstained tissue, in an unstained liver, in this case, I'll say liver. And what SSG does is very much a physics process. So there's an interaction of the photons with the tissue, and you get to see very fine collagen fibers usually being picked up that not even seen using normal stains like MT stain or PSR stains. And with that itself, you are then using AI algorithm, machine learning tools to be able to quantify the amount of collagen. The whole morphology and architecture of collagen could be quantified basically because of that. And that is very important, especially when it comes into NASH clinical trials, where you're measuring before and after treatment of the drug itself, what happened to the tissues, and all these important features and quantified data will be required to show if the drug works or not. And that, in terms of that, our tool, our AI platform, is being used as a secondary endpoint and in a FDA. And what it means as a secondary endpoint is that when the pharma or the biotech company, when they put in the protocol for their drug to be run on the trials, they use our tool to measure before and after treatment, and that is an endpoint. If they get to see that improvement using our tool, that means they have fulfilled their secondary endpoint, that endpoint, and that is something that FDA will take a look at it as well.
Okay, so you'd have specific algorithms evaluating NASH, or specific endpoints assessing the SSG in NASH, and then applying that to clinical trials. So then when you move into other diseases, it's going to be possible to use the same technology, but then just with different criteria, different scoring systems, and different AI algorithms?
That's right. You'll use a bespoke AI algorithm for specific disease of a different organ.
I see. And so as we move away, because I think you're probably not going to stay in the clinical trial space in this application, but as you move to more broader applications, what's the value proposition or the business case, so to speak, for this stain-free AI-based digital pathology system as you move away from clinical trials and into diagnostics? Is there a path forward there?
Yes. So we are quite different in a sense. We have validated technology. We publish easily about 10 to 15 papers every year just based on NASH and throughout the NASH work in the clinical trials. So you could just imagine with me the whole clinical trials in NASH, validate the technology that we have, validate the AI platform that we have. And when a drug has been approved, that validated platform and will then follow the drug all the way into the clinical diagnostic space, where we get to see how our technology could be used on a daily basis in the hospitals. So I can tell you that even on a regulatory side, we are pursuing a very much a FDA IVD pathway, towards both having both the hardware as well as the software going through clinical trials at this moment in time and getting approval for FDA IVD. And that is one of the routes that we are pursuing. So yes, there's a business case. Start off with the clinical trials, get it validated, having the pharmas and the biotechs involved in developing their drugs using our tool, and then moving towards the clinical diagnostics.
I think what makes that attractive is being able to deploy tools in clinical practice that were the same tools that were used to develop and validate to be used in the clinical trial, which is, I think, is very, very attractive. But then maybe a challenge or a downside is how practical is it going to be for a pathologist to use these tools in daily practice when they have, what we're looking to do is simplify or streamline the workflows. So is it going to be able to fit in to the daily practice, into the pathologist cockpit, so to speak?
Mm-hmm. Interesting. I think that's a very good question. And what we are, in fact, running a few trials right now on our own with the involvement of large pharma companies is to basically use our tool as AI aiding tool. And what it does is that it's a tool that aids the pathologist. You can look at it as an assistive tool. It assists the pathologist in terms of their daily workflow, in terms of how they scores and stage, for example, fibrosis. And what it does is that nothing is going to change on their side. On the pathologist side, they will still do the whole staging process of fibrosis using the same old way of categorical staging. But what is going to be different is that alongside right now, they have an AI tool that comes along with me, and that comes from SSG. They come from our readout from the AI part of it. It basically go alongside as the pathologist stage and diagnose, NASH. So that's ongoing right now. We are showing forth data to see how much more improvement with AI coming in to really aid pathologists as compared to without AI. So that's in a nutshell what we're going to do. And that's how we will foresee that going to be like, really integrating into the pathologist workflow on how AI could be used in the future. So for me itself, I will see that as that's how the future will be like, really, how machine and human will interact together, not replacing one another. I'm not saying that a machine is going to replace the human. I'm saying that the machine will really be helping the human in terms of efficiency, basically being more accurate, of course, in terms of and more consistent in the way the diagnosis is being done.
Okay. Yeah, I guess one of the beauties of, AI algorithms and AI technology is it can kind of be seamless. It's software. It can live in the cloud or even locally, really. So that might not be disruptive or cumbersome for the pathologist. That can kind of be running in the background. And maybe a good way to look at it is, this could replace the trichrome stain, right? Which we already use. So we obviously have different tools for different applications. But is there a hardware component that goes with this when we're measuring these SSGs or photons being emitted?
Yes. So you have to use specific hardware, and we have a hardware specifically for that. And that hardware will be placed into hospitals. That will be the hardware that will be used to scan and to get all your SSG signal coming out. So you're spot on. I think in terms of how it will be used, this will be quite a paradigm shift in terms of how we are moving towards replacing not the pathologists, but rather the stains, really empty stains, picosaurus stains, and these are the ones that we are seeking to replace.
Yeah. Well, Gideon Ho, thank you so much for being with us. So on a personal note, let's learn a little bit more about you. How did you come to be interested in digital pathology and artificial intelligence and developing these great tools?
Okay. I started off as an engineer. So I'm really a hardcore mechanical engineer by training. And I did my basic degree and honors degree basically in mechanical engineering. I moved on to get my master's in engineering in more like, I would say triple E, electronics and electrical engineering, and then PhD more towards bioengineering. So it all started when I was doing my PhD, how I was looking at the interaction of light with tissues. So laser tissue interaction, looking at collagen, and it all started during my PhD days. I then went on I did my postdoc, and that furthered my interest in this area. So my co-founder together with me, we started a company, and we wanted to use laser and how we could use laser in the area of looking at tissue diagnostics. That was how it all started. And of course, naturally, with the images that's being acquired using SSG, second harmonics, the next thing will be to use machine learning on this and to really quantify the biomarkers within this tissue. So we started looking at quantification before words like machine learning and artificial intelligence became the buzzwords. Those were like 10 over years ago. We started to quantify using rudimentary algorithms at the time. So that's how we got into AI. So it wasn't called AI back then, but it was, AI.
Yeah. Sounds like it's been an exciting journey, and I think it must be very gratifying to see it come together now to make it a reality in 2021. Yeah, like you said, AI is a very hot topic in digital pathology, and I think it's really going to change how we do things. So what excites you? Where do you see the field headed in the next 10 years or so?
What excites me definitely will be in the area of how the journey that we have. And every day it's really exciting when we see what we are doing, what the tools that we are developing being used today in the real world, in developing drugs for a very big problem in NASH. And how do I see that happening in 10 years? Today, we are doing clinical trials. Today, we are coming out, assisting the drug companies come up with a drug that will be approved by regulatory bodies. And what we will see in the near future will be how our AI digital pathology StainFree tool will be used in clinical diagnostics in hospitals throughout the world, and how it will be used to look at not just liver fibrosis, but fibrosis in different organs, in different diseases, including cancer.
Oh, wow. Not just liver fibrosis, but different organs, different diseases, cancer, and other applications. Well, our guest has been Dr. Gideon Ho from HistoIndex. We'll see you next time on Digital Pathology Today.
Thank you.
Thank you.
This has been Digital Pathology Today. Please be sure to subscribe. Thanks for listening.